Okay, welcome. My name is Andreas Baumbach. I'm an interventional cardiologist in London, and I am here with my co-chair to talk about what is a healing targeted drug-eluting stent and how is it different from a normal stent. Patrick.
Yeah, thank you very much, Andreas. I'm Patrick Serruys. I'm in the Galway at the University of Ireland, and I'll try to answer the question of Andreas. I mean, we are going to talk to you about the HT Supreme, and H3 is stand for Healing Targeted Drug-Eluting Stent, a new class of DES focus on rapid healing. It's quite amazing. It is based on a technology called electrografting, which is ultra-thin base layer covalently, that means very strongly bonded to the metal, to the surface of the stent. It acts as a blood metal barrier to eliminate the ion release and interaction. And it's very conformal and has a uniform surface which acts as a biological Velcro. I'll explain that a little bit later because Velcro is coming of two French words, velour and crochet, yeah? And that surface is very thin, as you could see, 200 nanometers, so it's really on top of the metal. And this coating is very important because without this electrocoating, you can have delamination of the coating, fracture, peeling, and that's really avoided after the electrografting coating, even if you push the device in a tortuous path track test and expand the device with the balloon. It is quite remarkable. On that cartoon, you see the metal at the bottom, and then you have this colorful substance, which is a monomer solution of polybutyryl methacrylate. And then if you put an electric current in that surface, you have a stretching of these molecules. They get aligned perpendicular to the surface of the metal in a kind of brush-like structures. Is only 200 nanometer, and that baseline coating facilitate the attachment of the polymer, polylactide, glycolide coating, and you see it's interdigitated in these baseline coatings. So it's really a top technology. So what is remarkable with this technology that the polymer will degrade within 60 days. That's the HT Supreme. If you think about the synergy, it's already 3-4 months, or the Orsiro, it's 13 months. And of course, you have the permanent coating. If you look at drug release, you can see in vitro that the release is quite fast. I mean, 28 days, if you look at the graphic, HT in yellow, orange, compared to the curve of elution of the science and the resolute. Drug dose is 1.2 microgram per millimeter square. The whole strategy or philosophy behind that strut elution is not to give too much cytostatics drug to the reconstruction to prevent the reconstruction of the neointima without blocking the endothelialization of the device. And then the peak of the drug, which is 10 microgram per gram, is really at 80 days, a critical moment when you have the massive migration and proliferation of the smooth muscle cell. And then looking at the orange curve, the drug dropped below a therapeutic level. If you compare that to the synergy, you see that the drug stay much longer above a therapeutic level, an inhibitory level. So we want to have something short, powerful, but leaving the same very quickly. And the result of that is the Evans blue is a dye 40,000 kilodalton, and if it goes through the endothelium, if the endothelium is permeable to the dye, the stented region will become blue, and that's a sign that the endothelium is not healthy. And you see in the mid portion, you have a bare metal stent, which is quite wide, is a little bit blue. And if you look at the upper panel, you have the Ealing Supreme, which is a little bit more blue, but not really permeable to the Evans blue. And at the bottom, you have the Xions. And in terms of histology, you could see that the HT Supreme has a pretty nice endothelium cover, and the connection between all the cell, that's technically the VE cadherin, is pretty good, as well as the protein 120. Andreas, it's your turn to talk about the clinical trial, a very important one.
Thank you. Yes, well, I think from this overview, we see that there are now several thousand patients in trials with the Supreme, from the first in man to recently the Pioneer. The Pioneer 3, the biggest study we call a Morecambe study, in 1,629 patients who were randomized 2-to-1 between the Supreme and a member of the Xions or Promus family. Patients could be included with several lesions, up to 3 de novo lesions in up to 2 vessels, and the lesion had to be covered with 1 stent. That was the inclusion exclusion. And within that, we then had a follow-up at 1 year, with the primary endpoint being the target lesion failure at 12 months. This is already the result. You can see 5.3 and 5%, a very low target lesion failure rate in both groups, and absolutely no difference. Non-inferiority was proven in this trial. We also found a very favorable safety profile with numerically less death and target vessel MI. That was, of course, not statistically significant because of the low numbers. There was no significant difference between the groups in terms of ischemia-driven target lesion revascularization. You can see again here on the left the combined endpoint, death or target vessel MI, not different, but numerically lower in the Supreme group. And then in the stent thrombosis, there was no difference. Again, very low rates of late stent thrombosis in the Supreme group. So overall, very good performance, clearly equivalence to the current market standard, and a very good safety profile.
Very good. Thank you very much, Andreas. So let's finish that conversation about the trial design of the Pioneer 4, which is going to start very soon. And I think there is 3 important components in this trial. First of all, thanks to Pioneer 3, we don't have to prove anymore any superiority of non-inferiority with the best in class. It has been done in the Pioneer 3. So we can come back to somewhat the mainstream of interventional cardiology. On one hand, what we call an all-comer trial. That means all kind of syndrome, stable angina, but unstable angina, acute coronary syndrome, STEMI, non-STEMI, name it. Secondly, all comers means also all kind of lesion, might be main stem, might be bifurcation, total occlusion, in-stent restenosis. All kind of lesion can be included, bypass graft, etc., etc. And the third point, it is an unrestricted use of the stent. 30 sites in Europe. And then the second key point of the trial, on one hand in red, we use the HT Supreme in both arms. In the red arm, we have an angio guidance and usual care. That means you use basically your subjective visual assessment, the high ball to say, I'm going to treat that lesion and spare that one. But in the blue arm, we are using QFR. And as you know, QFR is a way to judge the lesion physiologically, but it's derived from angiography. You got this beautiful, colorful angiography, and what is the transition between yellow and orange has to be treated. So that's the second big component of the trial. And finally, we embrace the concept of monotherapy. At the bottom, you could see that only aspirin will be given for 1 month. So you have a dual antiplatelet therapy with ticagrelor for 1 month, then ticagrelor for 11 months alone, and then you can go back to aspirin. And then it's very basic. The primary endpoint is patient-oriented composite endpoint, all-cause death, stroke, any myocardial infarction, and any clinically and physiologically driven revascularization. That's it. So I think it will be a very exciting trial. Andreas is one of the PI, and we'll try to start as soon as possible.
Exciting future ahead. I think I'm very excited about the Pioneer 4 trial. That will enlighten us as to the use of QFR on the one hand, and also give us more data and safety data for the Supreme.